Good morning to everyone. Myself Dr. T. Arun Kumar from the Department of Chemistry, Design Thinker, SNS College of Technology, Coimbatore. Today we are going to see about the metabolic pathway of TCA cycle. For definition, TCA cycle, that is the tricarboxylic acid cycle, is a series of enzyme catalyzed chemical reaction that form a key part of aerobic respiration in cells. This cell is also called the Krebs cycle and the citric acid cycle. The cycle consumes acetate in the form of acetyl coenzyme A and water, reduces NAD to NADH and produce carbon dioxide as a waste byproduct. The NADH generated by the TCA cycle is fed into the oxidation phosphorylation, that is electrotransport chain, that is a pathway. In eukaryotic cells, the citric acid cycle occurs in the matrix of the mitochondria. So this is cycle. So glucose 6 carbon, it is a glycolysis, and that is a 2-pyruvate we can generate in the glycolysis pathway. So pyruvate is converted into acetyl coenzyme A, then acetyl coenzyme A is converted into citrate. Citrate is followed by isocitrate. Isocitrate is followed by alpha ketoglutarate, then succinyl coenzyme A, succinate, fumarate, malate, oxaloacetate. Then again it is converted into citrate with the help of acetyl coenzyme. This is a cycle. So reaction of citric acid cycle. First one, citrate synthase, that is formulation of citronelyl coenzyme A intermediate, that is oxaloacetate is converted into citrate with the help of citrate synthase. Synthase is an enzyme which help conversion of oxaloacetate into citrate. Next one, accotinase is an enzyme reaction, that is this enzyme catalyzes the isomerization reaction by removing and then adding back the water to yield isocitric acid. So that is citrate is converted into isocitrate, the help of addition and removal of water. Then third step, isocitrate dehydrogenase. In the first oxidation step of the Krebs cycle, isocitric acid is oxidatively decarboxylated to alpha glutaric acid. That is, isocitrate is converted into alpha ketoglutarate. Here, NAD is converted into NADH plus H plus and removal of carbon dioxide with the help of isocitrate dehydrogenase enzyme. Next one, alpha ketoglutarate dehydrogenase pathway. This is a complex of different enzymatic activity similar to pyruvate dehydrogenase. That is, alpha ketoglutarate is converted into succinyl coenzyme A. Here also NAD is converted into NADH plus H plus. Carbon dioxide removes with the help of addition of coenzyme A. With the help of enzyme is called as alpha ketoglutarate dehydrogenase complex. Then fifth step, that is a succinyl coenzyme A synthetase. Succinyl coenzyme A, like acetyl coenzyme A, has a thioester bond with a very negative free energy of hydrolysis. That is GTP plus ATP is converted into GDP plus ATP. That is TP indicates a triphosphate, DP indicates a diphosphate. So GTP is converted into GDP and ADP is converted into ATP. That is adenosine diphosphate is converted into adenosine triphosphate. That is energy is liberated. That is succinyl coenzyme A is converted into succinate with the help of enzyme called succinyl coenzyme A. Here also coenzyme A is removed. The help of enzyme is called succinyl coenzyme A synthase. Then step 6, succinate dehydrogenases, that is oxidation of succinate to fumarate. This is the only citric acid cycle enzyme that is tightly bound to inner mitochondrial membrane. So we can know that the enzyme which is bound to the intermitochondrial membrane, that is inner is the same thing, it is present. So with the help of that only, this conversion of succinate to fumarate, that is FAD is converted into FADH2 with the help of enzyme called as succinate dehydrogenase. Then step 7, fumarase. The fumaric acid reacts with the molecule of water to form malic acid in the presence of the enzyme called as fumarase. That is fumarase converted into malate, the enzyme called as fumarase. The step 8, L-malate dehydrogenases, oxidate of malate to oxaloacetate. It is an NAD plus dependent enzyme. That is malate is converted into oxaloacetate. That is with the help of NAD is NADH plus H plus. The significance of TCA cycle: intermediate compounds formed during Krebs cycle are used for the synthesis of biomolecules like amino acid, nucleotides, chlorophyll, cytochromes and fats, etc. Intermediate like cyclinyl coenzyme A take part in the formation of chlorophyll. Amino acid are formed from alpha ketogluteric acid, pyruvic acid and oxaloacetic acid. Krebs cycle release plenty of energy required for various metabolic activities of the cell. By this cycle, carbon skeleton are got which are used in the process of growth and maintaining the cells. Conclusion: TCA cycle ATP generation, total ATP is 12 ATP. 3 NADH plus gives 9 ATP, 1 FAD gives 2 ATP, 1 GTP gives 1 ATP. So reference from the Leninger I referred. Thank you.